Chapter 16, our last and final chapter of Bio 130. And of course, it's going to be the funnest chapter, the reproductive system. We're going to go over four major concepts today: the male reproductive system, female reproductive system, menstruation, and mammary glands. And of course, what is the entire purpose of the reproductive system? To produce offspring. In order to produce offspring, we have sex organs, which we call gonads. In males, these are called the testes, and in females, we have the ovaries. And what these gonads or these sex organs produce are sex cells, which we call gametes. In males, we have sperm, and in females, we have eggs, which we also call ova, ovum, so this is singular, ova is plural, or oocyte, which we also call singular. This is a female gamete. What is the purpose of these gametes? When we look at these gametes, they each contain 23 chromosomes, and they come together through this process of fertilization to create a zygote. So you get 23 chromosomes from your mom and 23 chromosomes from your dad. From this 23 chromosomes, all of these cells are going to undergo mitosis, cell growth, and development, and eventually it's going to form you as an individual. So by the time you are a full-grown adult and the zygote, you are considered to be diploid because you have 46 chromosomes. Here, each of this gamete, whether it's an egg or a sperm, these are considered haploid because you only have 23 chromosomes. This process of creating these gametes undergoes what's called meiosis, and this is very different than how we create other cells in our body, which occurs during mitosis. So only sex cells, meaning those gametes, undergo meiosis. And why is this? This is because we create genetically unique cells. And if you notice, your siblings, you look completely different from your siblings, and that's because of all this genetic uniqueness. However, you do have some DNA overlap because, of course, you share the same parents. Now, if we look at here, we're going to take this as an example. This is a DNA from, let's say, your mom. And if you notice your genetic material from your mom's going to have a copy from her mom and also from her dad. Now, in this first phase of meiosis, we're going to duplicate this DNA. So we're going to create an exact copy of that DNA. So here's this original strand, and here that I'm making darker is a duplicated copy of this exact same strand. Now, this is how we can have what's called unique genetic material and genetic identity, and this is what's called crossing over. So this right here is called crossing over. And by doing this, we can eventually create 4 haploid cells that are genetically different from each other. So here we have this duplicated, this is chromosome, we'll say number 1, and this is chromosome number 2. Part of meiotic division number 1, we're going to duplicate all of this DNA, and we have crossover. So this region over here can cross over with this region over here. From here, we're going to have the first cell division. So this cell division, we're going to divide these cells in half. Okay. And then under the second meiotic division, we have these duplicated cells, so we're going to divide this in half one more time. So cell division 2, or meiotic division 2, this is going to result in 4 unique haploid cells. And of course, here we give an example of only one type of chromosome. So you can say that this chromosome has the characteristics for your hair color, your eye color, and that can be from your mom. And this chromosome has the characteristics of hair color or eye color from your dad. But only one of these are going to be expressed. Now with meiosis, you're going to pass on these characteristics and traits into your sex cells. Okay, so how we can create these 4 unique haploid cells are going to be different in females than in males, and that's what we're going to get into. So we're first going to start off with this male reproductive system, and the production of these gametes in the male reproductive system undergoes what's called spermatogenesis. And so the function of this is to produce your sperm cells. This begins at puberty as early as 10 years old and as late as 14 to 15 years old. It'll also continue throughout your life. And so males, unlike females, there is no end to the reproductive cycle. So they can have children as old as possible, so even up to 70 years old. All this occurs within the seminiferous tubules. Let's now take a look at the different structure of the male reproductive system. Here we have the testes. Inside of that house is the seminiferous tubules, right over here. Above this we have the epididymis. This long tube we have is this ductus deferens. Here it's going to empty into the ampulla of the ductus deferens, ejaculatory duct, and then we have the urethra, which is separated into three different regions depending on where it is. So we have the prostatic urethra, membranous urethra, and then the spongy urethra. And then both urine and sperm is going to be expelled into the external urethral orifice. Now the way I remember the pathway of sperm from maturation to release through the penis is by the mnemonic 7-up. So you'll never think of 7-up the same way again. Okay, starting off with S, this is for the seminiferous tubules, and this is where the sperm is first produced. It's then emptied into the ready testes, which is the R down here. And then sperm maturation occurs right above the testes at the epididymis. During ejaculation, we're going to have peristaltic wave-like contractions along this ductus deferens that moves all that sperm along. And then both the sperm is going to combine with secretions from the seminal vesicle and get released into the urethra through this ejaculatory duct. From here, ejaculatory duct, that sperm and secretions are going to get released through this urethra. So that's the U. Out the urethra and then out of the penis for P. Okay? So S, seminiferous tubules, ready testes, E being that epididymis, V being that ductus or vas deferens, E is being that ejaculatory duct, N, oh this one, I couldn't think of anything for that, so you can just think of it as nothing. And then U is the urethra and then out of the penis, so 7-up. Now what exactly is secreted alongside the sperm? So here from the seminal vesicle, it also secretes this thick yellowish secretion, in which case 60% of it is composed of that semen, and it has fructose, vitamin C, prostaglandins, and other substances that is going to nourish and activate all of that sperm. There's a cross-section of that male reproductive system. So here once again are the testes, epididymis, vas deferens. Here we have the ampulla of the vas deferens or ductus deferens. Here's a seminal vesicle that's going to give rise to the secretions to help activate all that sperm with the vitamins and the prostaglandins. And then finally out through this ejaculatory duct into the urethra and then out of the penis. Now other secretions that we have this bulbourethral gland, and this is going to secrete the fluids that's going to cleanse the entire urethra to get rid of that acidity from the urine and ensure that they have a decreased acidity once it enters, the sperm enters into the vagina. Let's now take a look at how we can produce sperm through the process of spermatogenesis. It's going to undergo under two parts. We have first part is meiosis, and then we have spermiogenesis. A couple of things, where does this occur again? We have this at the seminiferous tubules. If we take a cross-section of the seminiferous tubules, this is what we will see. We have the seminiferous tubules here in red, and then in everything in yellow, these are called interstitial cells. Other terminology you want to know is this inner area of the seminiferous tubule. This is called the lumen. And as we grow and divide and mature these sperm cells, it's going to go from the most peripheral region of the seminiferous tubules towards the lumen. So by the time we create all these mature cells, it's going to exit through the lumen, go into the ready testes, and then begin to mature here within this epididymis. Okay. Now, before we have, from birth to puberty, we have this stem cell line called spermatogonium, so the precursor to forming the sperm. And what the spermatogonium undergo is simple cell division, so it undergoes mitosis. And it's going to grow and divide and produce more spermatogonium cells. So from birth to puberty, all of this outer cell layer, they're just going to produce more and more and more spermatogonium cells. Now during puberty, we have a rise in follicular stimulating hormone, which we call FSH. And do you remember which region of the brain this is secreted from? Good. It's from the anterior pituitary gland. So during puberty, we have a surge of FSH, and this is going to cause our spermatogonium cells to grow and divide into two separate cells. We have a daughter cell type A, and we have a daughter cell type B. Now this daughter cell type A, this is going to remain within the basement of our seminiferous tubule as a stem cell. So it's just going to repopulate the spermatogonium stem cell population. So this eventually over time is going to keep on growing and dividing to form another daughter cell A or a daughter cell B. Okay, so it's going to keep on regenerating that A line. Now B, on the other hand, daughter cell type B, this is a cell that's going to eventually go under meiosis, and this is going to give rise to the genetic diversity for the sperm cell. Okay, so let's go over how that occurs. First, here's a quick illustration. Here's another cross section of the seminiferous tubule right over here, and we take a portion of that. Here we have that spermatogonium. Remember, it's diploid. We haven't undergone meiosis just yet. So it's going to undergo mitosis, which mitosis, remember, being the exact same type of genetic information. It's just going to grow and divide. It's going to produce its type A cell and then its type B cell. From here, type B cell, this is what's going to undergo the division and have unique sperm cells that's going to result in. Okay, so let's go over how that forms. So with this first part of meiosis 1, which is the first part of spermatogenesis, we have this daughter cell type B, and from here this is going to go form the primary spermatocyte. From this primary spermatocyte, this is going to go meiosis. So meiosis 1, remember, this is where we have, at this primary spermatocyte level, remember we have that from that illustration from before, we have a copy from your mom and a copy from your dad. Here it's 2N, meaning that one's from mom, one's from dad. This is going to be duplicated. And at this level, before meiosis 1 is complete, this is where you can have crossing over. So I'm just going to clear this out and we can show you with another color what this looks like. So it's much easier to understand. So here's red. So these are duplicated from mom and this is from dad. With crossing over, these two copies can cross over. So here's now from mom. And this portion has a piece from dad, so it's going to just cross over. Now, at the end of meiosis 1, what are we going to result in? We're going to result in secondary spermatocytes. So this is going to be one secondary spermatocyte, and this is going to be another secondary spermatocyte. Okay, each of these are now haploid because it only has one copy from mom and one copy from dad. Now, the second phase of meiosis 2, what are we going to do? We're going to cut this in half. We don't want to give both copies to our offspring, nor do we want to give both copies from dad to our offspring. So meiosis 2, now we have, this is that chromosome. Let me get my red. Okay, so here's that unique genetic material now. Okay, so here's 1, 2, 3, 4. So now we end up with 4 haploid spermatids because now this has 23 chromosomes, 23 chromosomes, 23 chromosomes, and 23 chromosomes. Okay, so as meiosis is occurring, we're going to push all of these spermatids towards the center of that lumen. Now, at the end of this meiosis, we form those spermatids, but they're not yet functional. And so we begin the next phase of spermatogenesis, being spermiogenesis, in which case we're going to start off with these spermatids. They have excess cytoplasm surrounding them. They're not motile, and they don't have a functioning tail. So this process of spermiogenesis, we're going to remove all of this cytoplasm and create 3 different regions. We have the head, the midpiece, and the tail. And this entire process is going to take 64 to 72 days. Now, by the end of spermiogenesis, we have the mature sperm, All of them are streamlined and equipped with a high rate of metabolism and a high rate of propelling itself. Because imagine, the sperm have to make its way from that seminiferous tubules through that ready testes, into that epididymis, down that ductus deferens, into the ejaculatory duct, down that urethra, into the vagina, through that uterus, into the fallopian tube. So it needs lots of energy and a means of propelling its way towards that egg. Okay, so if we look at this anatomy of a mature sperm, we have those three regions: a head, a midpiece, and a tail. And this tail is also called a flagella. So flagella is this tail structure that we have in the mature sperm cell. And this is the only place we find a flagella in the human body. And that's found within the sperm. This head, it contains this little helmet that you see here in red, and this is called an acrosome, which contains lots of digestive enzymes that are similar to lysosomes, so that the moment it reaches the egg, it can start digesting away the outermost layer of the egg, penetrate into the egg, and release this DNA genetic material into the egg. So here the head as well, it also contains all the genetic material. So here in purple is all of that DNA. This midpiece contains lots of mitochondria. For what reason? Good for that ATP production. And you remember, when it combines with that seminal fluid from that seminal vesicle has lots of fructose, it has vitamins, all that's needed to give energy to this midpiece to produce all of that ATP to allow the propulsion of this tail to move through that entire reproductive system of the male as well as the female. Now let's look at the different types of hormones and the effect that they have on sperm maturation. The first important hormone that we have in the testes is going to be testosterone, and that's produced by all these interstitial cells surrounding those seminiferous tubules. And this is activated by luteinizing hormone during puberty. From that on, testosterone is going to be produced continuously. Why do we need testosterone in males? It's going to stimulate reproductive organ development. It's associated with growth spurts, underlies sex drive. It causes secondary sex characteristics, including deepening of the voice, enlargement of the larynx, increasing the growth of hair, enlargement of skeletal muscles, and thickening of bones. Once again, all associated with that growth and development of the body. Let's take a look at these different hormones that cause and initiate the production of sperm. And so here we have initially the gonadotropin releasing hormone. This is produced by the hypothalamus. And remember from chapter 9, hypothalamus can regulate the actions of this anterior and posterior pituitary gland. So GnRH, this is going to cause and stimulate the anterior pituitary gland to release follicular stimulating hormone and luteinizing hormone. Once again, luteinizing hormone, this is going to trigger and cause the interstitial cells to produce testosterone. This is going to give rise to the maturation of secondary sex characteristics. But also we have follicular stimulating hormone, which is going to stimulate all those spermatogenic cells to produce sperm. Remember, you have the spermatogonia that's going to form that cell A, cell B, and then type B is going to form eventually all of these spermatids. And then for these spermatids to be finally functional and mature, we need this testosterone for that. So testosterone is needed for this final maturation of all the sperm cells. Now, the moment that we have too much testosterone, what's going to happen? We're going to send a negative feedback towards the hypothalamus saying that we have too much testosterone. That in turn is going to shut off this stimulus to the anterior pituitary gland. And by doing that, anterior pituitary gland is also going to decrease the amount of luteinizing hormone and FSH sent towards the maturing sperms. And so, of course, over time, what's going to happen, we're going to have a decrease of testosterone again. That's going to send a signal to our hypothalamus that we don't have a lot of testosterone. And then in turn, it's going to increase the secretions, once again, to the anterior pituitary gland and increase luteinizing hormone. So remember, we also need that balance of those hormonal systems for homeostasis. Okay, so let's take that one step at a time. I know that's a lot of information. So gonadotropin-releasing hormone is going to stimulate your anterior pituitary gland to release both follicular-stimulating hormone and luteinizing hormone. Follicular-stimulating hormone is going to target the spermatogonia, stem cells. And this is going to cause the stem cells to form either those daughter A cells or those daughter B cells. Daughter B cells are eventually going to form those secondary spermatocytes and primary spermatocytes, as well as those spermatids. Luteinizing hormone, this is going to trigger and target, the interstitial cells to secrete testosterone, and testosterone is going to be needed for the final maturation of all of those spermatids. Finally, with high amounts of testosterone, we're going to eventually send that signal to tell our anterior pituitary gland and our hypothalamus to say, hey, we have lots of testosterone, shut off that signal, and then finally, both hypothalamus and anterior pituitary gland are going to decrease the stimulus for the LH and the FSH. Earlier I mentioned that men can produce viable sperm to a very old age, and I was not kidding. Here is Ramajit Raghav. His claim to fame is that he's the world's oldest dad at 94 years old. Crazy, look at him. Still looks good, huh? Smile. And his baby is right over here. He's from northern India. Crazy, right? 94 years old, still having babies. Now, what's also interesting is if you do have children at an older age, studies have shown that you can also pass on longevity. And what do I mean by that? So let's take a look. Here is our chromosome in our condensed state of our DNA. And at the very tip of our DNA, our chromosomes, we have these capping proteins called telomeres. And these telomeres, their main function is to protect our genetic information. Every time your cells grow and divide, we give off our DNA itself gets shorter and shorter and shorter. And to protect our DNA, that's why we have these telomeres. So these telomeres here in yellow, they don't encode anything important within our body. However, as our cells grow and divide, these are going to get shorter and shorter and shorter until finally you start digging into the blue, which is your DNA. And why is that bad? Well, when we start digging into our DNA, then that's when you can give rise to diseases. In this specific case where if these genetic sequences, if they encode something important like proteins for metabolism or proteins for sustaining your cell replicative abilities, then that's when it becomes dangerous. Now, how is this different in sperm? Well, sperm, for studies have shown that the older you are, the longer your telomeres are going to be in your sperm. And by passing on longer telomeres in your sperm, that's how in turn you can pass longevity. So you imagine, here's someone, I'm just going to clear this off really quick. Here's someone who's a normal age. Let's say that this person is 40 years old, male. And these are the telomeres. And this is a sperm telomeres, or the chromosomes from that. And then here is someone who is, let's say that Ramajit Raghav. If his telomeres are this long, imagine how much more time it's going to take, how many cell divisions and cell cycles it's going to take in order for it to actually reach and make mutations within your own DNA. So it takes much longer. So longer telomeres means that the longer you live. Let's now move on to the female reproductive system. A couple of structures. We have the vagina. Here we have the uterus. Separating the uterus from the vagina is the cervix. And here we have the cervical canal. Out here we have the uterine, also known as the fallopian tube. And these finger-like projections surrounding the ovary, these are called fimbriae. This first region where our oocyte will enter into our fallopian tube, this is called the infundibulum. Inner lining of the uterus is called endometrium. And this is going to be the area that sloughs up every month and gives rise to menses. Myometrium, that's this inner layer. This is a muscle, smooth muscle layer. And then periometrium is this outermost region of the uterus. Here's a cross-section of the female reproductive system. And remember, anterior to the vagina, we have the urethra and the urinary bladder. So it's not in the same region as our reproductive system. So here we have the vagina. Here we have the cervix, the uterus, and then here we have the fallopian tube, ovary, and then those fimbriae. And what's the purpose of the female reproductive system is a production of ova, also known as oocytes, and this is going to occur within the ovaries. And what's interesting about the female reproductive system is that at birth, the total supply of all the eggs a female can release in her entire lifetime will already be present. So before birth, our cells are growing and dividing and growing and dividing. And then finally, when we can release eggs, it's going to start at puberty. And our reproductive ability is going to end at menopause. Another term you should know is menarche, and this is the first time a female is going to have her menses. Let's take a look at this process of oogenesis where we produce female gametes. We're going to start off with our stem cell population being oogonium. And from here, our cells are going to grow and divide through that process of mitosis to produce primary oocytes. So remember, in males, we have spermatogonium. Here, we're going to have oogonium. These primary oocytes are going to be embedded in the tissues within the ovary to form primary follicles. So here in the center is that primary oocyte. This tissue surrounding it is going to be those tissues from your ovary. Now, at birth, we're not going to have any more growth or development of these oogoniums. So at birth, you're going to have all of your primary follicles in place. And you'll actually have 2 million within your ovaries. So here we're going to have all these primary follicles. And then over time, we're going to have them inactive until puberty. And what sets off the maturation of these primary oocytes is the follicle-stimulating hormone that's secreted by our anterior pituitary gland. Now, this follicle-stimulating hormone and also the changes that occur with maturing the primary oocytes, the growth and development of the endometrium is all going to constitute the ovarian cycle. During puberty, you're only going to have 250,000 oocytes remaining, and a small number of those oocytes are going to be activated each month. However, only one oocyte is going to be matured and ovulated each month. So only 500 out of your potential 250,000 are going to be released into your fallopian tube in an entire lifetime. Now let's go back to see what I'm talking about. Here you have all of your primary oocytes, and while each month you're going to have that FSH, it's going to cause the maturation of all these primary follicles. You're going to grow and divide and grow and divide. However, one of them is going to mature and outmature the other primary follicles, in which case that first one that matures is going to then be released by the ovary and enter into your fallopian tube. And that's also why you only get 500 out of all of these 250,000 that are going to be released into that fallopian tube. Reproductive life of a female, not like a male's, it's going to last until 40 years at best, and around age of 11 to 51 is when you have your reproductive lifespan. As I mentioned earlier, the maturation of these primary follicles is going to be stimulated by FSH, or follicular stimulating hormone. And so with an increase of follicular stimulating hormone, these primary follicles are going to grow and mature and grow and mature until finally it forms a mature vesicular Graafian follicle. And what's characteristic of this stage is we have this fluid-filled chamber called an antrum. At the same time, as these primary follicles grow and grow and grow and mature to form this mature vesicular follicle, it's also going to give off the hormone estrogen. And this estrogen, once that estrogen peaks, it's going to stimulate ovulation. And so what we're left off with is the secondary oocyte, which is going to be the one that's going to enter into that fallopian tube. What's left over, this follicle that's left over, it's that ruptured follicle, eventually it's going to form that corpus luteum, which is also necessary that we need to know about this because this is going to produce another hormone called progesterone. And we'll see why that's important in a little bit. Now, at the same time, as this primary follicle grows and matures, it's also going to begin the process of meiosis. Here we have this primary oocyte. It's arrested in prophase 1. So the prophase 1 of that cell cycle. So from that primary follicle to finally it's a mature Graafian follicle, it's going to just grow and mature until finally this cell is going to undergo meiosis 1, in which case it's going to form what's called a first polar body and a secondary oocyte. Remember, the secondary oocyte, this is the form that's going to be that oocyte's going to take place during ovulation. Now, this first polar body, this has genetic material in it. However, this genetic material is not going to be the one that's going to form you as a zygote. So this is going to stay on the surface of that body, so it's going to stay within that ovary. However, this form, the secondary oocyte, this is what's going to then be released into that fallopian tube. Now this is where we're going to stop. So if and only if we have maturation, or we have fertilization of the secondary oocyte, do we proceed to meiosis II. In which case, this secondary oocyte will come together with a sperm. We're going to have meiosis II, in which case we're going to form an ovum. An ovum is a fertilized egg. And this is also going to form a secondary polar body. And this polar body is going to form 2 more polar bodies. So let's do that breakdown really quick. So if fertilization occurs, we're going to eventually end up with 3 polar bodies and 1 ovum, which is that fertilized egg. No fertilization. We're arrested in the first part of meiosis, in which case we only have 1 polar body and 1 secondary oocyte. We'll get into this in a little bit more, so I know it sounds a little confusing because it's a lot of terminology at one time. Okay, now ovulation of that secondary oocyte occurs with the release of luteinizing hormone, and that secondary oocyte is surrounded by, so here this primary oocyte, we had those tissues surrounding it to form that primary follicle. Here we have this ovulated secondary oocyte. Now we have tissue surrounding it, but it's not the same from our ovary. This is called corona radiata. So it's like a nice cell capsule surrounding that secondary oocyte. And here is this corona radiata surrounding our secondary oocyte over here. So it's kind of that nice halo. Okay, here's that overview one more time. So meiosis II is completed after ovulation if and only if the sperm penetrates that secondary oocyte, meaning that we have fertilization. Fertilization, once that ovum is formed, that 23 chromosomes from that mom can be combined with the 23 chromosomes from the sperm to form a fertilized egg, in which case we have that zygote, which is diploid and has 46 chromosomes. If no fertilization occurs, secondary oocyte is not penetrated by sperm. Eventually, the secondary oocyte is going to die and does not complete meiosis to form an ovum. So it's going to be arrested in meiosis I. Let's do a quick review of oogenesis, starting off with our oogonium stem cell population. It's going to be diploid and undergo mitosis. Exact genetic information, no genetic diversity from your parents. Here, this is going to grow and divide to form primary oocytes. These primary oocytes are going to be suspended in the tissues of the ovary to form primary follicles. At birth, you have 2 million primary follicles that are identical to each other. Now, during puberty, you're going to have the follicular stimulating hormone. It's going to cause this primary follicle to mature every month until it's going to grow and mature to form a mature vesicular follicle, also known as a Graafian follicle. Prior to ovulation, we're going to divide this mature vesicular Graafian follicle into a first polar body and a secondary oocyte. Now this is where we're going to stop. So secondary oocyte, this is going to be what's released by the ovary into that fallopian tube. And if fertilization happens, the sperm comes into contact with a secondary oocyte, then we're going to complete meiosis, in which case we're going to form a second polar body and an ovum. This first polar body is also going to grow and divide to form 2 additional polar bodies. If fertilization does not occur, we're going to stop over here and we're only going to have 1 first polar body and 1 secondary oocyte. I'm going to clean this off really quick. I'm going to go over this one more time by looking at the chromosomes because sometimes that gets pretty confusing. With this diploid, before birth, you have a copy from your mom and you have a copy from your dad. Now, this is going to be found here within your primary follicle. Then over here, as this primary follicle grows and divides, we're going to undergo meiosis 1, in which case we're going to duplicate the genetic information from both of your parents. So your mom's and then your dad's. And then during this phase, this is where you can also have that genetic diversity, meaning that you can have crossing over where your mom can get some of your dad's and your dad can get some of your mom's. Here we have the secondary oocyte that's formed. So at the end of meiosis 1, we have this polar body and the secondary oocyte. So this is going to form first polar body, and here is your secondary oocyte. So secondary, and that's your first polar body. Now this is, again, this is a stage that's arrested unless you have fertilization. So if fertilization occurs, then we're going to split this genetic material one more time. And so then we have a second polar body, an ovum, and then 2 additional polar bodies. So 1, 2, 3, and 4. And of course, these are all going to be genetically unique and not identical. So these can be the polar bodies, and this is that ovum. Okay, more Guinness Book World Records. Youngest mother, Lena Medina, at 5 years old, 5 years and 7 months old, she was pregnant. And it's pretty sad because the children get raped by family members, so she actually was impregnated by her uncle. But on the flip side, the other oldest lady, who is a mother, is Amkari Panwar, also from India. And she's 70 years old when she had a baby, so she had twin babies. So, crazy, right? Still people, yeah, old age, still going for it. Okay, now let's take a look at the uterine or menstrual cycle. All this occurs within the uterus, and this is a cyclic changes of the hormones that are going to help the maturation of that endometrium, that inner lining of the uterus, to prepare the body for implantation of that fertilized egg. Okay, so let's go over this. It occurs under 3 different phases. We have the menstrual phase, proliferative phase, and then we have the secretory phase. So here is an overview of the different stages, the menstrual phase, the proliferative, and the secretory phase. And we can notice that this maturation of this endometrium is always going to correspond to hormonal levels for estrogen and progesterone. We're going to start off with the first phase of the menstrual cycle being the menstrual phase. And this is when that functional layer of the endometrium is sloughed off. So it's going to detach from that uterine wall, and this is what we know as that menses, so the bleeding. It's going to bleed for 3 to 5 days, and all in all, women expel 50 to 150 milliliters of blood during that period. Okay, so let's see that. This is the very first phase. So menstrual flow is going to be during this phase, and the endometrium, because we're flowing off, we're removing a lot of the inner lining. A lot of this endometrium, the functional layer, is going to be low. It's going to be decreased because a lot of it's sloughed off. If you look at hormonal levels, we don't have a lot of estrogen and we don't have a lot of progesterone. Primary follicle is right over here. And because our primary follicle, now we're starting the new phase, by day 5, now we're going to start secreting hormones to mature the next primary follicle. So primary follicle, you can see here, primary follicle follicular stimulating hormone is going to start increasing to start maturing primary follicle to that secondary follicle to eventually we have that vesicular follicle. Second phase is proliferative phase, and this is when we're going to start regenerating that endometrium. So we're going to have an increase of estrogen levels from those growing follicles. We saw that earlier. And as the endometrium increases, we have more cells, more tissue, so what do they need? They need a nutritive supply of blood and oxygen. So you also increase your blood supply to these layers. And because of that, your endometrium is going to become thick again and well vascularized and ready to prepare for implantation of that fertilized egg. At the end of this proliferative phase, you have ovulation, in which case you're going to have an increase of luteinizing hormone. Let's take a look at that over here. So here we have that primary follicle. It's going to continue to grow and mature and grow and mature. As that happens, remember, it secretes that estrogen, and that's why we see this increase of estrogen levels rising. And of course, with estrogen increasing, we also increase the proliferative layers of this endometrium. Eventually, at the very end of this proliferative phase, we have a surge of luteinizing hormone, which is released by the anterior pituitary gland. That's going to cause ovulation. So here we have ovulation. With ovulation, you're going to have the formation of this corpus luteum. Corpus luteum is going to be maintained by the luteinizing hormone. So luteinizing hormone is going to maintain this corpus luteum. And remember, this is going to start producing the hormone progesterone, which is why during ovulation, now we have this corpus luteum. Corpus luteum is going to start increasing the levels of this progesterone. And progesterone is going to then in turn have another role to maintain this endometrium. Secretory stage, we have levels of progesterone released by that corpus luteum is going to start rising. It's going to increase the blood supply to the endometrium, causing the glands to increase in size and increase nutrients to that uterine cavity. And of course, once again, we're going to prepare that endometrium for implantation of that fertilized egg. If fertilization does occur, the embryo is going to produce a hormone that's going to cause the corpus luteum to continue producing progesterone. So what this hormone is HCG. We saw that in chapter 9, human chorionic gonadotropin hormone, and this is going to take the place of that luteinizing hormone sent off from the anterior pituitary gland. So this is going to cause corpus luteum to continue to secrete progesterone and continue to maintain that endometrium. If fertilization does not occur, we don't have any signals from this HCG. Corpus luteum eventually is going to deteriorate and degenerate as LH blood levels start declining. If you don't have any LH, then what you're going to do, you're not going to have any blood vessels supplying the endometrium. Those cells and tissues within those endometrium is going to eventually die, and then you're left with menses. Let's look at that over here one more time. Here's that corpus luteum that has now just formed during ovulation. Luteinizing hormone is going to maintain this corpus luteum, and that's going to allow the secretion of progesterone. And that's why we see this increase of progesterone levels rising. And then here we have this increase of endometrium layer. And of course, if we have implant fertilization here within this endometrium, then that's going to take over. HCG secreted by that developing embryo is going to take over instead of this luteinizing hormone, and that's going to continue the production of progesterone during the first trimester. Other things, so how does progesterone and estrogen work? We can think of it like a brick building in which estrogen is kind of like the bricks forming the building itself, but progesterone helps keep all these bricks stationary in place. So that's how they work collaboratively together to maintain this endometrium. Okay, one more time going over these hormones. Estrogen is going to be produced by those growing and developing follicle cells, so from that ovary towards that Graafian follicle. And while that's occurring, it's also going to cause secondary sex characteristics. So development and enlargement of the uterine tubes, the uterus, vagina, and external genitalias. Development of the breast, widening of the pelvis, and also the onset of menses. Progesterone, again, this is produced by the corpus luteum once that secondary oocyte has then been ovulated. It's going to stop producing hormones 10 to 14 days after ovulation, in which case we need other mechanisms to take over. You need that human chorionic gonadotropin hormone to take over to cause the corpus luteum to continue to secrete progesterone. It does not contribute, important, to the appearance of secondary sex characteristics. It does help maintain that pregnancy to prevent contraction of that myometrium. Remember, myometrium is that muscle layer surrounding the uterus. If you have contraction of this later, that can cause sloughing off of that endometrium. And it also prepares the breast for milk production. What is important about birth control and how does this work? Well, it stops ovulation, and the pills contain synthetic forms of both estrogen and progesterone. You remember, the growth and development of all these follicles depend on, and as well as development of that endometrium is all going to depend on rising levels of estrogen and rising levels of progesterone. However, birth control, what this does is has synthetic forms of these two so that we don't have any peaks of estrogen and no peaks of progesterone. Therefore, we don't in turn have any release of that luteinizing hormone from our pituitary gland. Therefore, we have no release of that mature egg and we have no development of that Graafian follicle. So let's look at this one more time. Synthetic estrogen is going to stop that pituitary gland from producing follicular stimulating hormone and luteinizing hormone to prevent ovulation. It's going to support that uterine lining to prevent breakthrough bleeding mid-cycle. Progesterone is going to stop the pituitary gland from producing luteinizing hormone to prevent that egg from being released. Also, it's going to make that lining inhospitable to fertilize egg. Remember I told you estrogen builds up that wall, that endometrium. However, progesterone is going to help keep everything stationary in place. The synthetic form, it's not going to do that. It's not going to help hold everything in place, making it, this inner lining, very unstable. Now, I had a student one time asking, well, why is it that I'm taking birth control and my urine is very concentrated? And so let's look at this case study where an 18-year-old patient wants to take Yasmin, it's a birth control pill, but wants to know some side effects that can occur. The pill contains drospirenin, which is a synthetic version of the progesterone that your body naturally produces. During first half of the menstrual cycle, estrogen will produce water in your body, and the natural progesterone helps get rid of all that water. The synthetic progesterone and all other contraceptive pills is based on the testosterone and does not eliminate water. So what visible side effects do you have? Bloating. And if you don't eliminate water, what color will your urine be? It'll be more concentrated. So this is why a common side effect of taking these birth control pills is that bloating appearance. Now let's move on to the last topic we're going to cover, and that's the mammary gland. It's present in both sexes, but only functional in females, and it's also considered a modified sweat gland because it produces milk. Couple of structures. Here we have the nipple, the protruding central area of the areola. Areola being this pigmented area surrounding the nipple. Alveoli, this is where we contains all of the glands that produces milk, and it's then excreted through this lactiferous duct into the nipple. Other structures here, alveoli, once again producing all of that milk during lactation, produced and it's secreted into this lactiferous duct, into the sinus, and then we have the opening of the lactiferous duct within the nipple. Mammography, or a mammogram, why is this important? Well, it's needed to detect breast cancer in women, as breast cancer is the second most common cause of death in women. 1 in 8 women will develop this condition, and it's hereditary. 10% of it is hereditary. And as you remember, I was telling you about proteins and why are proteins so important and regulating these proteins within the body. Here we have two types of proteins, BRCA1 and BRCA2, and these are the most common proteins found to cause this type of cancer. Here we have that cell cycle. Remember, you're going to have different proteins activating that cell to either go cell division, mitosis, or to prevent mitosis from happening. Anytime your DNA is damaged, your cell is going to automatically undergo what's called apoptosis, which is programmed cell death. So anytime damaged DNA, your cell is going to die because, why is that? It's a protective mechanism to prevent any damaged DNA from continuing and causing things such as cancer. BRCA1 and BRCA2, these are two types of proteins needed to repair damaged DNA. However, if it's unregulated, these proteins can cause repair of damaged DNA, which in turn can give rise to mutations and specifically uncontrolled cellular growth. So BRCA1 and BRCA2, there is typically found an increase in these proteins, so you have increase in DNA repair when that cell wants to die and stop growing. Other common reasons why you get breast cancer and lifelong exposure to estrogen, whether you have your menses early or you have a late menopause, or if you have estrogen replacement, so estrogen replacement. Mammograms, so this is a X-ray to look for different lumps So, of course, being cancerous, you're going to have uncontrolled cellular growth, giving rise to lumps that you can feel within your breast. It's a good idea to always feel around your breast tissue, also within your armpit area, because remember, we have lymph nodes there, and anytime you have cancer cells, your own cells can detect them and also engulf them and bring them to a lot of these areas, those lymph areas, to start screening them and try to attack them. Recommended every 2 years for women between 40 and 49 years old, and yearly thereafter. And so here's this procedure of a mammogram procedure. Malignancy, so here's an uncontrolled cellular growth, and here's normal tissue. Okay. All right, so that's it for the first part of chapter 16, and I will see you in the second part.